Hi, I hope everyone is at the best under the pandemic condition that we are having right now around the entire world. Anyway, in this video lecture, we're going to, have an insight. We're going to learn and the virus that is causing widespread disease in humans around the world. Now, this particular video lecture has been divided into two parts. In the first part, we're going to discuss about, what are microorganisms, what is a virus, what is the composition of the virus. Then we're going to study about the immune system of human beings, how this immune system is going to fight against any sort of infection which is caused by a pathogen. And the second part of the video, we are going to discuss or we're going to study about and we're going to learn about the coronavirus, the virus which is basically causing the widespread infection in humans. And what are the steps we should take in order to prevent the spread of infection among human beings. What are the proposed the treatments that is available and that scientists are basically proposing. What is the possibility with the vaccines that is going to be made, or probably many companies around the world they're trying to make a vaccine against this particular virus. So we are going to discuss about coronavirus and its related issues in the second part. In the first part, we'll discuss about microbes and viruses and about the immune system of the human beings. So let's start. Now, what are microorganisms? Now, microorganisms technically these are microscopic organisms. That means they're so small that we cannot see with our naked eyes. We just cannot see any microorganisms with our open eyes. All right, maybe because of that, most of these microorganisms they can escape our sight and they gain entry inside our body. And once these microorganisms enter inside the body, we all know what is going to happen. That is going to cause infection. It starts from at one place and it starts spreading in our entire body. You know so uh, so what is the definition of microorganisms? Technically these are very minute organisms, microscopic organisms. Now in order to see these microorganisms, since we cannot use our eyes in order to see them, we're going to use special instruments. Okay, and these instruments are called as microscopes. So we have this microscope which is a compound microscope. Then we have another one which is scanning electron microscope. Now what is the difference between these microscope? It is the level of magnification. All right, so usually compound microscope magnification is little less. It is used in the laboratories, in colleges, in schools, where you can see a cell or maybe a bacteria under this type of microscope. Whereas your scanning electron microscopes are used, and these are very costly. They have high magnification, and which is usually used to study the structure of a cell, or in this case a particular microorganism. So we will be needing such kind of instruments in order to study a microbe, since we cannot see with our naked eyes. Now, the branch of biotechnology that deals with the study of microorganisms is called as microbiology. All right. Now, microorganisms are single celled. Like most of the microorganisms, these are single cells, but they are going to stay in a colony, just like human beings. Human beings, they always stay in colonies. So similarly, microorganisms, these are single celled organisms, all right, but they are going to stay in colonies. So in this particular picture, you will see a petri dish. And this is a petri dish where bacterial colonies are growing. Now you can see there are a number of colonies in it. Each of these colonies are going to have millions of these bacteria. Okay. Now, so each of these colonies are different from one another. Say, suppose this is a red colony, this is it is a yellow colony. So the organisms in these colonies, the bacteria in these colonies are also different. That's why they're forming different colonies. They have different colors of colonies, and that again depends upon the media. You have specific media that is allowing the growth of specific microorganisms, but they will again form colonies. Anyway, the concept is that the microorganisms are usually single cell, but they're going to stay in a colony. So they're going to form colonies. And this picture is a graphic representation. You can see single organisms here, okay? But they are all staying as together. So they're going to form a colony. So you have to understand that microorganisms, they always stay in colonies. Now let us discuss about virus. What is a virus, okay? Now here in this picture, you will see different examples of virus. You have types of viruses in it. You have bacteriophage, you have adenovirus, human immunodeficiency virus. This is quite famous among all HIV. Then we have hepatitis, we have Ebola that, was quite, they cause widespread infection, once upon a time, few years ago. We have influenza, adenovirus, papillomavirus, rotavirus, rabies, etc. Now you'll see they have different shapes also. Now Ebola is like this. Some others they have spherical structures like this. Some are having a polygonal structure like this. Okay, so We have different types of viruses, they have different shapes also, and different kinds of infections they're going to cause. Anyway, now what is a virus? Now virus is, again, it is a microscopic organism which usually causes dreadful infections in plants, in animals, in humans, and even in bacteria. All right. Now most of the scientists, they do not consider a virus to be a microorganism, because why? They consider that these viruses, they fall between living and non-living organisms, which means when they are outside, that means they are in the environment. All right. So they are non-living, they are not growing, they're not multiplying. But once they bind to a particular host cell means, like, say, suppose a cell or maybe an organism, it can be a plant, it can be animal, it can be human beings. So when they enter inside a particular host organism, they become living, and they can start spreading infection in that organism. So you can say when they are outside the individual or the organism, then they are non-living. And when they enter inside a living organism, which is called as the host organism, then they become living. So that's the reason why scientists do not use the term microorganisms with viruses. All right. So they consider that virus basically falls between living and non-living and organisms. Now the size of the virus is very small. Okay, they're very, very small. You can see the size ranges from 20 nanometer to 400 nanometer. And what is the scale here? 1 nanometer is equal to 10 to the power minus 9 meter. All right, so it is so small. All right, it is even smaller than bacteria. So the smallest bacteria is having around 400 nanometer. Okay, so imagine, it is so small. It is so small that it can even infect bacteria. Bacteria itself we cannot see with our naked eyes. It is smaller than those. And there are certain viruses which can infect bacteria. So this is one example, bacteriophage. Okay, okay, now viruses, as I said, when they are outside the host, they are usually non-living. But how then they're going to multiply the number, how they're going to increase its number? They can increase its number only inside a host organism. So they have to, in order to increase its number, they have to infect an organism. Okay, and those organisms are called as host organisms. If they are infecting a cell, then that cell is called as the host cell. If they're infecting an organism, the entire organism, like human beings, then we are called as the host organism. So how they're going to multiply and increase the number? They can multiply or increase the number inside a living cell, or inside a particular organism. So host organism or host cell, that is the term which is basically used. So they depend upon the host cell for carrying out its important functions. So that's why they are called as parasites. I hope everyone know what is this particular parasite. The meaning of parasite is when they are going to grow or on a particular organism, and they are not providing any help to these organisms. They're extracting everything out of the organisms, okay, and make the organism paralyzed, or maybe they're going to damage them. So viruses are regarded as parasites because they depend on other cells in order to carry out its functions. Now, artificially, if anyone wants to study on virus, we cannot grow virus on a very simple medium like we can grow bacteria. Now, one of these slides I showed you, one petri dish containing bacterial colonies. We cannot grow viruses like that. In order to grow virus, we need living cells, all right? Because in one of the point I said they can multiply or increase only inside the living cell. So we need to have a cell culture where we can grow these viruses and we can go for its studies. And obviously, depending upon the type of virus, the type of cells are also going to change. Now, what is the composition of virus? Now, when viruses, outside the host cell, they usually exist as independent particles, and these particles are called as virion. All right. Now, all viruses, okay, all true viruses, they will contain genetic material. Now, we all know, you have probably heard about DNA all the time. DNA is quite, uh, commonly used these days, okay. So all true viruses will contain genetic material just like us or the plants or the bacteria, but they will either have DNA as the genetic material or they're going to have RNA as the genetic material, not like us, because we have both. Plants and animals and human beings, they have both. They have both the DNA as the genetic material and they have RNA also inside the cell. But when it comes to your virus, a true virus will have genetic material, but either they're going to have DNA as the genetic material or RNA as the genetic material. Now, when they have DNA as the genetic material, those are called as DNA viruses. And if they have RNA as the genetic material, then those are called as RNA viruses. I have given you a few examples here. Like DNA virus, we have the smallpox virus. Everyone know what is smallpox, chickenpox virus, herpes virus. Okay, the viruses that causes these diseases, these are all having DNA as a genetic material. So these are called as DNA virus. Now we have RNA as the genetic material in some of the viruses. Those are called as RNA virus, like coronavirus. Now this is the most common virus these days, which is causing such a dreadful disease in humans, and that spreads so fast among human beings. All right, so coronavirus is a RNA virus. Then we have influenza virus, which was quite in the news a few years ago. Similarly, it was also causing respiratory diseases in humans. So influenza virus is also an RNA virus. SARS, it was again quite famous in the early 2000s, which was again causing a respiratory disease in humans. So this is also an RNA virus, and everyone knows about HIV. What is HIV? All right, HIV is the virus which is responsible in causing AIDS in humans. Okay, so all these viruses are RNA virus. Now, whether a virus contains DNA or RNA is a genetic material, these genetic materials can be your double-stranded. That means you're going to have two strands of DNA, or you're going to have two strands of RNA, which is joined together by bonds. So these genetic materials either can be double-stranded, or these genetic materials can be your single-stranded also. Or they can be circular, or they can be linear. Linear means it is going to be just like a thread, okay, long thread. And when it is circular, imagine circular means like, a spherical, the ends are basically joined together. It is not open. The ends of the particular genetic material is not open. It is basically joined. If it is open, then it is linear. If they're joined together, then it is your circular. So these genetic materials can be double-stranded or single-stranded, or they can be circular or linear. So this is the composition of virus. Now let's talk about what a particular virus has. Like for example, in this particular diagram, what we have. So we have already talked about the genetic material which is found inside the virus, which can be either DNA or RNA. Now this genetic material is always within a particular protein code. Can you see this particular structure? That is the protein code, which is called as the capsid. So genetic material is always confined within this protein code, which is called as the capsid. All right. Now this is a particular viral cell. Now some of the virus, they have these envelope around it. So those type of viruses are called as enveloped virus. Okay, so they have an envelope around the capsid, so which is called as the enveloped virus. Examples like coronavirus. Again, the coronavirus is very common these days. Everyone knows about it. So coronavirus is an envelope virus, which means you can imagine this is a coronavirus. Okay. Then we have retroviruses. HIV, is a retrovirus. Then we have paramyxovirus. All these viruses are your envelope virus, which means they have an envelope around the capsid. All right. Now, apart from this envelope, the most important things they have are these glycoproteins. Okay, these envelope glycoproteins, which is present on the surface of the envelope. Now, what is a glycoprotein? And glycoproteins is nothing but we have proteins and we have carbohydrates. So when they're going to join together, they form glycoproteins. So all these envelope virus, okay, all these envelope virus are going to have these spike-like things on the surface of the envelope. These are called as the envelope proteins or glycoproteins. And what is the importance of these, glycoproteins? Because of this protein, it is possible for the virus to attach to a particular host cell. Remember, I told you outside the host cell, they're non-living. Now, how they're going to increase their number? They have to enter inside a host organism. Now, once it enters inside the host organism, they have to start infecting, right? So the first mode of infection is what? They have to attach themselves to a particular cell. Now, how they're going to bind to a particular cell? They're going to bind to a particular cell with the help of these glycoproteins, which is expressed on the cell membrane, okay, on this envelope actually. So even coronavirus has the same structure. Okay, the coronavirus is going to have all these structures on the surface, which is going to make them, attached to a particular cell, and they can start causing infection. This attachment is important so that they can, penetrate the genetic material inside the cell. Then the whole process of infection is going to start. We will discuss about it anyway in the coming slides. Now, when we talk about, RNA virus which contains RNA genetic material, most of these RNA viruses, they have segmented genome, which means that segmented genomes means they are not going to have one single RNA. They're going to have more than one individual RNA molecules. All right. So because, some of the DNA virus, they will have one either single-stranded DNA or double-stranded DNA. Some of the RNA viruses, they are going to have one single-stranded RNA or one double-stranded RNA, but some of them, they can have more than one. All right. So such kind of viruses are going to have segmented genome. Now, the best example is the influenza virus. We have come across influenza virus in the earlier slides also. As I mentioned, influenza virus is going to cause respiratory disease in humans. Okay, and it was quite in the news a few years ago. Now, see here, they have 8. How many RNA molecules are there? There are 8 single-stranded RNA molecules. So that's why they have a segmented genome. Most of the virus that causes infection in plant, in animals and humans, these are your RNA viruses only. Most of them, okay? Not every virus is the RNA virus. Most of the RNA virus that causes infection in plants, animals and humans, these are having RNA as the genetic material. Now, some of the virus that contains RNA as a genetic material, these are termed as retrovirus. Now, why they call it? Not all, okay? Not all RNA viruses are called as retroviruses. Some of them are called as retrovirus because of the presence of one particular enzyme found in association with the RNA molecule, and that is called as the reverse transcriptase enzyme. We will discuss about what is the function of reverse transcriptase enzyme in the coming slides, but you have to understand that not all RNA virus are called as retrovirus. Only few of them are called as retrovirus because they have an enzyme, a very important enzyme, which is called as reverse transcriptase enzyme. So this is the structure of HIV. Now, HIV is a retrovirus, okay? So these things we have discussed already. We have the envelope, then these are the glycoproteins, then they have a capsid, and inside which they have the HIV genome, which is an RNA molecule only. And along with the RNA molecule, you're going to have, excuse me, certain enzymes in it, okay? These enzymes are called as the reverse transcriptase enzymes. Because of these enzymes, they are called as retroviruses. All right, then. So this is an example of HIV virus. Now, transmission of virus. Now, how virus is going to be transmitted from one organism to another. So when viruses are transmitted, they're basically transmitted with the help of vectors. Okay, now vectors, another term you can use for vectors as carriers. So virus is not going to travel like this. Someone has to, transmit a virus from one place to another. So it can be a wind, like say, suppose if the virus is, in the air, the wind is going to carry the virus from one place to another. Or it can be, individuals, like right now humans are basically spreading the coronavirus. One human can spread the coronavirus to other human beings. All right, it can be between animal or human, it can be between plant and human, it can be through insects also. Did you understand? So these are basically the carriers of the virus. Otherwise, virus is not going to travel on their own. They do not have any wings, so they will be carried with the help of vectors or carriers. Now, most of the plant viruses are transmitted through insects or other organisms that feed on them. That's quite common. Now, animal viruses, they usually spread through single-celled organisms, or from animals to humans, or from humans to humans. Now, I guess everyone knows that coronavirus was basically, they're trying to track down how this coronavirus came into human beings. And most of them, they are quite sure that the coronavirus basically came from bats to the humans. So animals to human transmission of viruses can be there. Right now, it is not a bat which is responsible. It is a human being which is transmitting the virus from one human being to another. So again, human to human also, virus can be transmitted. Now, certain, very dangerous disease that is caused in humans and animals, these are basically, carried by arthropods. Arthropods means insects, okay? So 8-legged organisms. So most of the dreadful diseases that occurs in animals and humans, these are carried by arthropods. Like, we have mosquitoes, we have ticks, etc. Examples of the diseases that is caused by these viruses, like dengue virus. Everyone knows what is dengue virus. It is still there in the news right now. Coronavirus is in the news, but dengue is still there. The Japanese encephalitis virus, so these things are always there. And these viruses are basically carried through arthropods only, like mosquitoes and ticks. Now, what is the mode or steps of viral infection? Let us discuss the step by which the viral is going to cause infection in an organism. So virus is going to increase the number of viral reproduction that we have learned already. Now, once the virus is inside the cell, okay, once the virus is inside the host cell, what is going to happen? It is going to produce the viral particles. It is going to cause the lysis of the cell, and it is going to go out. That means it is going to lead to the damage of the host cells. So basically, the virus is going to kill the host cell. But sometimes what is happening, take for example HIV. HIV, whenever it is going to infect a cell, it is not going to kill the cell, but it is going to functionally paralyze the cell. Did you understand? And once the viral particles are produced, they are going to leave the cell one by one. So basically it is not killing the cell, but it is going to make the cell functionally paralyzed. So that's what happens when HIV enters inside human beings, and that is how it is going to paralyze the cells of the humans. Okay. So there are two ways. Either they're going to cause damage of the cell, or they're going to make the cell functionally paralyzed. Let us discuss the steps. The first step is basically the attachment. I think in one of the slides I have already mentioned that attachment of the virus to the particular host cell is important. And what is making that possible? The glycoproteins that is present on the surface of the envelope, that is making it possible for the virus to attach to a particular host cell. So the first step is attachment. Second step is penetration. What is penetration? Now, in order to cause infection, they have to, penetrate inside the living cell. So either the entire virus goes inside the cell. Sometimes the entire virus doesn't go. Only the capsid, which is containing a genetic material, that is inserted into the host cell, or sometimes it is only the genetic material that goes inside the cell. Okay, so that is a penetration. So first is an attachment, second is a penetration. That means either the virus is going to enter, or the capsid is going to enter along with the genetic material, or it is only the genetic material that is going to enter inside the cell. That is the second step. What is the third step? Third step is your replication and assembly. Now here we have to discuss three very important points. Now, if it is a DNA virus, then what DNA virus is going to do, once the DNA genetic material of the virus enters inside the cell, it is going to take the help of enzymes and proteins of the host cell in order to make the viral mRNA. Then viral mRNA is going to give the proteins. These proteins are going to form the viral particles. Then these viral particles are going to lyse the cell and move out of the cell. So it can cause infection in the rest of the healthy cells which is present in the surrounding. So that is with the DNA virus. If it is an RNA virus, then again what the RNA virus is going to do, once the viral RNA is present inside the host cell, again just like a DNA virus, it is going to use the enzymes and the proteins of the host to make a viral mRNA, a copy of the RNA that they already have, a viral mRNA. And this viral mRNA is going to be translated into proteins. Then proteins are going to form viral particles. These viral particles are going to cause lysis of the cell, that means damage of the cell, and it is going to move out. Once it has moved out of the cell, it will start causing infection in the rest of the healthy cells which is present in the surroundings. So that happens with the RNA virus. In one point, I already mentioned that I also mentioned about certain RNA viruses which are called as retrovirus. Now, in such cases, what happens? Retrovirus is completely different. Okay. Now, if it is a retrovirus, then once the RNA molecule of the virus enters inside the host, what is it going to do? They're going to make a copy of the DNA. Did you understand? Using the viral mRNA, they're going to form a copy of DNA. And this DNA will enter inside the host genome, which is present inside nucleus, and it will get integrated. Okay, so integration of the retroviral DNA is important. So once the DNA has been produced, it will enter inside the nucleus. It gets integrated into the genome of the host, which is usually the DNA. And along with the genome of the host, they're going to be transcribed into mRNA. So in this case, they will produce viral mRNA. Okay, so the DNA gets inserted into the genome of the host. Then along with the genome of the host, they're going to form an mRNA, which is the viral mRNA. And this viral mRNA is going to be translated into proteins. Proteins will give you viral particles. Viral particles is going to cause, a lysis of the cell and go out and start spreading infection. So we have three different mechanisms of replication and assembly. Release, as I mentioned, so once the viral particles are produced, they're going to cause lysis. They will be released out of the cell. Once they're released, they will start binding to the rest of the nearby cell which is present in the surrounding. These are healthy, and they will again start the same process of viral infection. So these are two pictures here which is clearly showing how this viral infection is going. Say attachment, penetration, okay, then production, assembly, and they are going to be released. Now this is a picture of retrovirus. Okay, retrovirus, as I said, it is completely different. So we have the viral mRNA. Reverse transcriptase enzyme is going to help in the formation of a viral DNA that gets integrated into the genome of the host. They produce the viral mRNA. Viral mRNA is going to give us the proteins, which will be assembled into your viral particles. Then these viral particles are reduced by the cell. All right. So this is a retroviral infection. This is a normal viral infection. How it is basically going to happen. All the steps are basically shown here. Okay. And this is just a picture of different kinds of viruses which causes different kinds of diseases in humans. You can go through it later on. Now let us discuss about the immune system. This is the second part of this video lecture, immune system. Now what is immune system? The immune system is nothing but it is a network of cells and organs and various proteins of our body, which is going to fight against a pathogen once it enters inside the body. So once the pathogen enters inside the body, we have this whole network, okay? We have this whole network of cells, then, various molecules, which are proteins only. And we have certain organs also, which is going to work together in order to remove or kill the pathogen from the body, which means it is going to prevent the spread of infection, okay? Now, the main parts of the immune system includes white blood cells. We have RBCs. We have, those are called as red blood cells. We have white blood cells. We have antibodies. Then we have the complement system. Now, this system is entirely made up of proteins again. Then we have lymphatic system, which is associated with our tissues. That is our skin, the cells that we have below the skin, the spleen. Everyone, I guess, knows about the spleen. It's a very tiny organ which is present in the abdominal cavity. Then we have thymus, and then we have a bone marrow. Okay, so these are the main parts of the immune system. That includes white blood cells, antibodies, complement system, lymphatic system, spleen, thymus, and your bone marrow. Now, irrespective of the type of cells, all the blood cells are produced inside the bone marrow. Okay, and once they have been produced, they're released into the bloodstream. Now, once they're present in the bloodstream, what they do? Now, since blood is flowing in the entire body, every inch of our body is going to have blood. Blood is flowing. So once these, blood cells are released into the bloodstreams, they will start flowing. Okay, in all the parts of our body, they will look for pathogens. Now, once they find a pathogen, they will try to kill the pathogen so that they cannot cause any infection. Okay, so that is the work of these immune cells. And these immune cells, the blood cells are produced inside where? Again, it is produced inside the bone marrow. Then they are released into the blood. Now, these are the different types of WBCs. We have neutrophils, eosinophils, basophils, monocytes, T cells, B cells, natural killer cells, macrophages. All right, these are the important WBCs. All these cells are produced inside the bone marrow, as I have mentioned in the last slide. Now, out of all these, I guess very common are T cells and B cells. I guess everyone knows what is a T cell and B cell. These are collectively called as lymphocytes. So we have lymphocytes, which are, number one is a T cell, another one is a B cell. Sometimes natural killer cell is also regarded as a lymphocyte. Then we have certain cells which are called as phagocytic cells. Now, we have to understand what is phagocytosis. Phagocytosis very simply means eating. So cell is going to eat. That process is called as phagocytosis. Cell can drink also. Then it is called as pinocytosis. Okay. So phagocytosis is nothing but it is the mechanism by which a cell is going to eat. Now we have these immune cells. Okay. Number one is a macrophage. Then we have neutrophil. So they are phagocytic cells. Now what they're going to eat? They're going to eat the pathogen, and that's how they're going to remove the pathogen from the body. So they're going to eat pathogen. Okay. Neutrophils, macrophages, the eosinophils, they are going to eat the pathogen, and that's how they're going to remove the pathogen. Basophil is a very important cell which responds during allergic reaction. Anyone having any allergic reaction, can be skin allergy, food allergy, anything it can be. So all these allergic reactions are caused because of these basophils that we have in the system. Okay. So these are different types of WBCs that we have. Now, let's talk about, okay, fine. One thing I forgot about it. Let me tell you about one very important property of the immune system. What is that? Now, see, when the pathogen enters inside the body, our immune system has this very important property is that they are going to remember what pathogen has entered, okay? Like say, suppose you are exposed to a pathogen for the first time, our immune system is going to remember what pathogen they have entered. So that in the future, when the same pathogen is going to enter, our immune system is immediately going to kill the pathogen so that they cannot cause any infection. Is this clear? So it is a very important property of the immune system. What is this property? It is called as memory. Okay, our immune system has a very good memory. They're going to remember what pathogen has entered for the first time. So the second time, or maybe in the future, when the same pathogen is going to enter, they will immediately kill the pathogen so that the pathogen is unable to cause infection. Okay, and that property is called as memory. And that property is because of the presence of a type of cell which are called as the memory cells. Okay, so these memory cells are going to be present inside the body, which is going to remember what pathogen entered for the first time. So when it is going to again re-enter, this pathogen obviously will re-enter again in the future. So whenever it is going to re-enter, these memory cells are going to prevent your reinfection. So these memory cells will immediately recognize the pathogen, will kill the pathogen immediately, and it is going to prevent reinfection. Now, how these memory cells are going to perform this very important function, you have to understand the lifespan of the memory cells. Lifespan of the memory cells is like many years, like some are for 15 years, 20 years, some are for like lifelong. Imagine lifelong these memory cells are going to be present inside our body. And these memory cells are going to give us protection in the future with the same pathogen. And that is the basis of vaccination. Now I guess you have been hearing about vaccines of corona. What is a vaccine? Vaccine, whenever it is injected inside the body, this important cells need to be produced. So when vaccine is producing, injected inside the body, memory cells will be produced against the particular organism. And these memory cells, since it is present, since the lifespan of these memory cells is very long, so it is going to give us lifetime protection. Important cells during infections are the lymphocytes and phagocytes. Excuse me, I have already explained about lymphocytes, B cells and T cells. Phagocytes, I have told you about macrophages and neutrophils. Phagocytes means cell eating. Okay, so these cells are going to eat the pathogen and remove it from the body. So you can go through this particular picture later on. These are the different types of immune cells that we have inside the body and the type of functions they're going to mediate. Okay. Now we have two branches of immune system. One is called as the humoral branch. Another one is cell-mediated. Humoral branch is the humoral branch. We have B cells and antibodies. Cell-mediated branch, we have T cells and phagocytes again. So here, just look into this particular diagram. You have we have humoral immunity. So these are the B cells. So B cells, when they interact with an antigen, this is an antigen, they get activated. Okay. Once they get activated, they will differentiate into two types of cells. Some of the B cells will give rise to memory cells. Remember, we have come across memory cells. The lifespan of these memory cells are very long, and they're going to give us future protection against certain pathogens. Okay, so they're going to remember what kind of pathogens has entered in the first place. So B cell interacts with an antigen, they get activated, they will produce memory cells. Some of the B cells are going to produce plasma cells. Now plasma cells are the immediate cells, which means immediately they will produce antibodies, which means antibodies are the final products of a B cell. These antibodies will try to remove the pathogen from the body. So you have to remember like this, the memory cells are for the future and the plasma cells are for the present. So at present, we need protection, right? And who is giving this protection? These plasma cells are giving protection. Now, how these plasma cells will give protection? These plasma cells are going to produce these proteins. These proteins are called as antibodies. Antibodies are going to help in the removal of the pathogen from the body. So this is a humoral immunity where we have B cells. Then we have cell-mediated immunity where we have T cells and we have phagocytic cells. About them, I have already explained you in the earlier slides. Anyway, so T cells, we have two populations T cell. One is a TH cell, another one is a TC cell. They interact with the pathogen, they get activated, and they're going to remove the pathogen from the body. Now TH, okay, H stands for helper. What is the function of helper? Helper means it is going to help. Now what is the helping nature they have? This T cells, TH cells, when they get activated against the pathogen, they will help in the activation of other immune cells, which includes your B cells. They can activate, see, they can activate B cell, they can activate TC cell, they can activate the phagocytic cells. Did you understand? So T helper cells are very important. So once T helper cell gets activated, it is going to activate other immune cells. Now our immune system is ready to fight against the infection. Did you understand? Then TC cells are basically going to destroy a particular target. So can you see this? This is the infected cell. They have caused destruction. That means this particular cell has been destroyed completely from the body. So this is a cell-mediated immunity where we have T cells and your phagocytes. This is a humoral. Next, we have two types of immune response. One is called the innate immune response. Another one is adaptive immune response. Innate immune response means it is the first line of defense, like we have skin, we have enzymes in the body, and these are present since birth. Whereas your adaptive immune response, we acquire this type of immune response. And how do we acquire this type of immune response? Only when we are exposed to a pathogen. Did you understand? Only when we are exposed to a pathogen, then we acquire the immune response, and that is called as adaptive immune response. And that is basis of vaccination also. Remember, I told you about memory cells and all. So memory cells are produced during adaptive immune response when we're exposed to a pathogen. And these memory cells, as I mentioned before, they're going to give lifelong protection. So these are the cells which are present in the innate immunity, and these are the cells which are present in adaptive immunity. So you have to remember B cells and T cells. These are the major cells of the adaptive immunity, and these are the major cells of the innate immunity. Okay. Lymphocytes, as I said, there are two types. One is TH, it is called as helper because it helps in the activation of other immune cells. Then the next one is a TC, C stands for cytotoxic. B cell interacts with an antigen, produces antibodies, so antibodies are the products of a B cell. And what is the function of antibody? They will help in the elimination of, or neutralize the pathogen that has entered inside the body. Antigen is nothing but it is a pathogen. It's a foreign object that has entered from outside. Now, so this is the most important function of T cell. What is the function of T cell? It helps in the activation of other immune cells. Okay. Now, activation of T cells again depends upon phagocytic cells. Now, important phagocytic cells includes macrophages, dendritic cells. Okay. These two cells are very important for the activation of a TH cells. So once TH cell gets activated, they will activate B cells, they will activate TC cells, they will also activate macrophages. Did you understand? So you can see there's a link between all these cells. One cell is activating another cell, that cell is activating another cell, and the third cell is activating the first cell. Did you understand? So there is a link. There has to be some sort of communication between them. Then only they are going to get activated together, and they're going to together fight against the infection, the pathogen that has entered inside the body. Okay, so activation of T cell depends upon what? The phagocytic cells. So these are the list of phagocytic cells. And spleen is an organ which is going to remove any sort of pathogen which is present in the blood. Now pathogen can be present in the tissues, pathogens can be present in the blood also. So pathogen which is present in the blood are basically removed in the spleen. Okay, so spleen is a very tiny organ, as I mentioned in one of the slides. It is present in the abdominal cavity, and the importance of spleen is that it is going to remove any pathogen that has entered into our blood. Okay, and that's all. So this is the end of the first part of the video. So in the second part, we'll discuss entirely about coronavirus. Thank you so much.